So what happens over here is whenever there is a secondary structure of protein, we say, okay, means whenever there is a folding or coiling of the primary structure, then at that time, two forms of structure, secondary structural proteins are formed. Okay. So when there is a secondary structure formed, in this you will find there are again two forms. One is alpha helix structure and second one is the beta pleated sheet. Now, what is this alpha helix structure and the beta pleated sheets? Alpha helix means nothing but it's a structure which is formed obviously due to the folding and coiling of the primary structure, wherein the size group of the R is large. Okay, in the alpha helix structure, what happens is whatever the size group of the R side chain is there, that side is there, it will be large. Whereas in case of the beta pleated sheet, the size of the R group is small. So this is the first difference between these two structures. Okay, second difference is now obviously the in order to form the alpha helix, it should be bounded by something, right? So here there comes the action of what? The intermolecular H bonds. Means H means the hydrogen bonds. And this bond is formed between the CO and NH. Means the CO of the, say, first amino acid and the NH of the fourth amino acid. So whenever this helical structure is formed, there happens to be a thing called as a twisting. Okay, helical means obviously you can say that there is a twisting, right? I'll draw it over here. This is how the helical structure is there, alpha helix. So these twists are there, right? So this twist always you will find that it is in right-handed form. Right-handed screw form. Forming a particular shape which is called as the helical shape. So in a secondary structure of the protein, if there needs to be formed alpha helix, what should happen over there is there will be a intermolecular hydrogen bond formed between the first amino acids carboxyl group and the fourth amino acids NH group. Okay, and whatever the helical is there, that twisting will be in the right-handed screw form. This is about what? The helical structure. And the best example of the alpha helix is nothing but the keratin. Now, what is this keratin? It's a protein which you will find in nails or say hairs. Okay, so this is the first structure, which is alpha helix. So beta pleated sheet. Now, first point, as I already told you, that is the size group R will be small in the beta pleated sheet. So in the name itself, it is there, pleated sheets. Okay, whenever there is a folding or a coiling, it is going to form what? Some kind of pleated sheets. So in this, what will happen is there will be an what? Intermolecular bond formed between, again the same, CO of one chain with NH of another. But the only difference what will take place over here is that here there will be helix formed, here there will be sheets formed, and the sheets are formed in such a way that they can slide over one another. The sheets can slide over one another. So because these sheets are capable of sliding over one another, it happens to be given an appearance of what? 3D structure. So you'll get 3D beta pleated sheet. I'll just draw again the diagram, so it'll be easy for you to understand. Okay, these kinds of sheets will be formed. Beta pleated sheets will be formed. Okay, and one of the example which I can give you over here is silk. Silk is the best example of what? Beta pleated sheet structure. Okay, so this is what happens in what? Whenever a protein undergoes the secondary structural changes. Now, what will happen when it enters into its tertiary structure? Now, tertiary means what? 3D structure. So there will be a 3D appearance when the secondary structure further undergoes folding. 3D structure when there is a further folding of secondary structure. So when there is a three-dimensional structure you get, okay, due to the again means refolding of the secondary structure, it gives rise to two basic types of protein. Okay, so one is called as the this one globular protein, and the other one is called as fibrous protein. Okay, so it gives rise to what? Two forms of protein. That is, one is the globular and other one is what? The fibrous protein. Okay, now usually what happens is whenever a three-dimensional folding, I mean a three-dimensional structure is formed due to the refolding of the secondary structure, there is a stability which has been obtained. Okay, I'll just write it down. Okay, because of this 3D structure formation, what will happen is it will stabilize the secondary as well as the tertiary structure of protein. But in order to get this stability, it will require some kind of binding between them or bonds formation between them. So there are few bonds which I'm going to write over here, which can form what? A 3D structure, which can give rise to a 3D structure. So the first one is hydrogen bond, the common one. Second is disulfide linkages. Third is van der Waals forces. Fourth is electrostatic force. And fifth is hydrophobic interactions. Okay, so whatever secondary structure, tertiary structure is there, most probably the tertiary structure, okay, that structure gets its stability by using these bonds, that is hydrogen disulfide, van der Waals forces, the electrostatic force of attraction, and the hydrophobic interactions. Okay, so now whenever this tertiary structure is formed, now as I told you, right, either there will be one polypeptide or there will be more number of polypeptides depending upon how many amino acid residues are going to bind to it. So suppose if there is one polypeptide, then that particular thing is termed as what? Monomeric. And if there is more polypeptides, means more than one, then it is called as oligomeric. So suppose if there are more than one polypeptide present, they will be called as what? Oligomeric. Oligomeric means what? These are the peptides called as what? Protomers. Okay, so now what is this protomers? Okay, protomers are nothing but such kind of peptide bonds, or sorry, peptide chains, which will be held by weak forces. Okay, if the chain is very long, so obviously what will happen is the weight will be more, and there are chances of it to get broken up. So therefore, whatever the forces which is going to bind this huge chain, okay, that will be held by what? The weaker force. So because of that, what is very important over here is the stability. Means you need to get what? The native state of the protein. Now, what is this native state of the protein? Native state, original state, right? Native state, when that protein will gain is whenever there is a normal pH and temperature. So if that particular polypeptide chains, or say that protein, if it is having a normal pH as well as the temperature, then whatever the shape of that protein is there, it will be more stable. Shape will be more stable. Okay, so if that particular is for particular protein is what, shape specific. Okay, if that particular protein is what, shape specific. Shape specific means what? It is having all the conditions proper, that is normal pH and normal temperature, then that state is called as native state. It's called as what? Native state. Then the fourth structure, which is the quaternary. Now, obviously, in the quaternary structure, you may have understood what is a quaternary structure by now. It's nothing but whenever there is a polypeptide chain formed, okay, by two or more times of folding, or what do you say, this one, twisting, then that is called as what? Quaternary structure. So two or more polypeptide chains, they come together, it's okay, they will form something called as subunits. Okay, whenever two or more polypeptide chains come together, they are known as what? Subunits. And whenever there is a spatial arrangement of the subunits, we call it as what? Quaternary structure. Yeah. The last point is, see, now I told you, right, in order to stabilize the protein structure, there are some forces or there are some bonds formed, right? So I am just explaining you what actually that bonding means. Okay, just few bonds I'll be explaining. First one is, I'll take hydrogen bonding. So hydrogen bonding means what will happen is, these forces will be operated between, what do you say, a positive and negative atom. Like for example, because it's a hydrogen bonding, means what? This force is going to form a between bond between partially positive hydrogen and partially negative atom. Okay, so such kind of forces which takes place to form a bond is called as what? Hydrogen bonding. Then second one is hydrophobic bonding. Now hydrophobic bonding means what? Now the R chain is there, right? The R side means the side chains. These side chains in the amino acid will be hydrophobic sometimes. Phobic. Phobic means what? Against, fear of. Means hydro means water. Okay. So when some of the side chains in the amino acids, which amino acids? In the same amino acids, okay, the same polypeptide chain, what happens is the side chains will be hydrophobic. So at that conditions, what will happen is, means in the aqueous solutions, the proteins, they will fold to give a clustered inside folds. So whenever this clustered inside the folds happen, then there will be two, this one is the polar side chain, which will be hydrophilic in nature. And this hydrophilic will be where? Outside, means on the external part, outside structure. Okay, one more time I'll tell you. Hydrophobic bonding. See, hydrophobic means what? Water hating, right? So sometimes what happens is on the same amino acid, whatever the side chains are there, they will be hydrophobic in nature. So in an aqueous solution, whatever these proteins are there, they will undergo the folding. But in this folding, what happens is whatever the inner folds are there, right? There they will be hydrophobic, whereas the outer ones, which is there, the outside structure, they will be hydrophilic. Means the inner folds will be water hating and the outer folds will be water loving. Okay. And the last type of bond, okay, an important bond is the covalent bond. Very similar type of bond. Now, covalent bond is a bond which occurs between what? The S atoms of two residues between two adjacent chains, called as the covalent bonds. Okay, between the S atoms, whatever it is there, like the disulfide linkage, right? S double bond S. This is called as what? Disulfide linkage. Di 2 sulfide S, right? So these things are called as what? Covalent bonding. Okay, so if a protein structure needs to get stabilized, these are some of the forces which acts on it. Okay.